Hello, I wrote a paper for chemistry. Somebody wants it, I'll be happy to send it to them. I'm thinking I did pretty good with it. I'd like to read as much as I can, or at least summarize, but in this paper I'll show the present hypotheses on the origin of biomolecules, that there are credible scientific experiments to support these. Some of the problems will also be presented to show that researchers are working on solving currently. This paper, in the hopes of this author, should be informative and interesting. The origin of biomolecules on Earth. What we know is that even the first cell would have required chains of macromolecules that, no matter how long, would have required extremely small steps. The first would invariably be the simple molecules bonded with carbon. Interestingly enough, carbon is not the only element that this works with, as diatoms use silicon instead of carbon, that continue to comprise our cells and give us our only similarity with prokaryotes. The first experiment that gave the research on biogenesis I can't pronounce that, was Uri Miller. A triumph for science in this experiment. Stanley Miller showed that biomolecules could arise from a mixture of hydrogen, methane, ammonia, and water in a series of tubes to simulate conditions on the early Earth. The original experiment produced 5 amino acids, but recent reanalysis has shown that 22 amino acids, 5 amines, and many hydroxylated molecules were actually produced. This process's first mixture was from these basic compounds to hydrogen cyanide, formaldehyde, and other compounds such as acetylene and cyanoacetylene. After that, through Strecker synthesis, amino acids were formed. These are the basic components of every protein in the human body. Here are the chemical formulas. I put a complicated chemical formula after that. Next paragraph I put down. Okay. Sorry, I thought it wasn't recording. Next paragraph is not really chemistry, but more. Recent studies. I have this down in the bibliography if somebody wants to look at it. Recent studies show that in old parts of our DNA have the presence that these parts are filled with the amino acids readily made in the Urey-Miller experiment. Other than the obvious, this is amazing evidence for the theory of common ancestry according to Oxford. To understand to quote Oxford, to understand more fully how amino acid composition of proteins have changed over the course of evolution, a method has been developed for estimating composition of protein in an ancestral genome. Estimates are based upon the composition of conserved residues in descendant sequences, and empirical knowledge of the relative probability of conservation of the various amino acids. The Urey-Miller experiment was simply using a 5-inch, I'll skip that part since I'm sure everybody knows. With the success of this experiment, some people started to think that all that was needed was a good shake and life would jump out. Let's skip that. Next experiment was Joanne Oral I. Florenza was the next big name in the origin of life research, and her biggest contribution was the synthesis of the nucleobase adenine, which was the missing key component from the Urey-Miller experiment, from hydrogen cyanide, which was one of the missing purines from the original experiment. She used several assumptions, mostly focusing on weather and temperature of the early Earth. Most importantly it was uniformly hot, experienced ice ages, and had ice caps, which I don't think that's too far-fetched, and how the solution became concentrated, water freezing, leaving behind pure ice crystals and an organic brine that would become ever increasingly concentrated. This experiment was done by slowly freezing a flask of dilute hydrogen cyanide to negative 20 degrees Celsius to get a solution of hydrogen's. It was been said together with the Urey-Miller experiment that's fundamental results of prebiotic chemistry. These findings with a concentration of hydrogen cyanide caused several more independent experiments with other compounds. And one such experiment is that of Leslie Orgel. Orgel used concentrated formaldehyde to produce a rich variety of sugars and ribose. This also led to the longest experiment in the origin of life studies, that of Stanley Miller himself. By now, he was considered the Einstein of this field. He redid the Leslie Orgel's experiment, but this time put the samples in a freezer for 20 years. When the samples were brought out for examination, they found an abundance of adenine. The amino acids we could not synthesize enough of this. Had been elusive till now and put another metaphorical brick in the wall. These experiments, as good as they were, had some major... Oh, I'm at 6 minutes. I'll skip that part, but I talk about some of the problems with it. In dealing with these problems, a couple interesting places that included biomolecules, macromolecules, and even cells. Several places were thought to include deep sea, deep crust, and deep space. Now that several places have been described, I will talk about extremophiles and a little about their part in general. The first was to the bottom of the sea, where to the amazement of everybody there were animals living without even ever having seen the sun. This was found to be a veritable treasure trove of specimens. The sample ranged from crabs to 8 feet long tube worms with no stomach. The tube worms were most notable for being able to survive extreme temperatures, 130 degrees Celsius at the head to negative temperatures at the tail. And with no stomach, this creature deserves a research lab of its own. All of these living on single-celled algae which lived on a form of photosynthesis that instead of relying on sunlight relied on chemical imbalances caused by undersea vents. This was chemosynthesis. I am at 7 minutes. Okay, the experiments for deep sea vents have been done by many, but I would like to talk about one in particular that was done by Robert M. Hazen. These are called the gold tube experiments, which were to subject a plausible prebiotic chemical mixture of water, carbon dioxide, and minerals to a controlled environment. As the name suggests, a gold tube. The first set of experiments, he sealed pyruvate with carbon dioxide to produce biomolecules beyond the scope of this paper. Preceding experiments have heralded similar results. Okay. I'll stop it there and go to the next one.